Welcome back to the deep dive. Today, we're going super small. Like smaller than you can even imagine.
Smaller than an ant.
Way smaller. We're talking about the Krebs cycle.
Ah, the Krebs cycle. Fundamental metabolic pathway.
You got it. And it's happening inside every cell in your body right now.
That's right. It's happening inside the mitochondria.
The powerhouses.
The powerhouses of the cell.
Okay, so this Krebs cycle, what does it actually do?
Well, you eat food, right? And your body, it takes that food and it turns it into energy.
Magic.
Basically. Well, not magic, but a very elegant process.
So the Krebs cycle turns food into energy.
Not directly. It's more like a middleman.
A middleman.
Yeah, it extracts energy-rich electrons from the food and then it passes them off.
Passes them off to who?
To another process called the electron transport chain. That's where the bulk of the energy is made in the form of ATP.
Okay, so the Krebs cycle is like setting the stage for the main event.
Exactly. It's all about setting up the electron transport chain, which we'll talk about later.
Right. So where does this Krebs cycle actually take place?
Like I said earlier, it all happens inside the mitochondria.
Those tiny factories in our cells.
Precisely.
All right, I'm ready for a factory tour.
Okay, but before we get into the cycle itself, there's a transition phase.
A transition phase.
Yeah, remember glycolysis? That process that breaks down glucose outside the mitochondria.
Right, I think I remember that.
Well, at the end of glycolysis, you end up with pyruvic acid.
Pyruvic acid, and then...
Then pyruvic acid gets converted into acetyl-CoA.
Acetyl-CoA, that sounds important.
It is. Acetyl-CoA is like the key.
The key to what?
The key to the Krebs cycle. Without it, the cycle can't start.
Interesting. Okay, so we got our acetyl-CoA. What happens next?
This is where the cycle begins.
All right, let's do it.
So first, acetyl-CoA, which is a 2-carbon molecule, combines with oxaloacetate.
How many carbons does oxaloacetate have?
4 carbons.
Okay, so 2 carbons plus 4 carbons.
Equals 6 carbons. They join together to form citrate.
Citrate, like the stuff in oranges.
Well, not exactly, but it's related.
Okay, so 2 molecules merge to form citrate. And how does that happen?
There's a special enzyme that makes it all happen called citrate synthase.
Citrate synthase, all right, got it. So we've got citrate, what's next?
Next, citrate gets rearranged into its isomer isocitrate.
Isomer.
It means they have the same atoms, but they're arranged a bit differently.
Like rearranging furniture.
That's a good way to think about it.
So what's in charge of this molecular rearrangement?
Another enzyme called aconitase.
Aconitase. They all have such interesting names.
They do. All right, step 3. This is where things get interesting.
Okay, tell me more.
Isocitrate gets oxidized.
Oxidized.
It means it loses electrons, and when it does, it releases a molecule of carbon dioxide.
CO2. Like we breathe out.
Exactly. And those electrons that are released, they're picked up by a molecule called NAD+, which turns into NADH.
NADH, isn't that an energy carrier?
You got it. NADH acts like a shuttle. It carries those high-energy electrons to the electron transport chain.
Where they'll be used to make ATP.
Exactly. And the enzyme that makes this step happen is called isocitrate dehydrogenase.
Isocitrate dehydrogenase. Wow, that's a mouthful.
It is. So we've lost some CO2. We've made some NADH. What happens to our original molecule?
I don't know. You tell me.
It's been transformed into a 5-carbon molecule called alpha-ketoglutarate.
Alpha-ketoglutarate, that's the star of step 4.
You are correct. Step 4 is a lot like step 3.
Another oxidation.
Yep, alpha-ketoglutarate loses another carbon dioxide, and NAD+ picks up more electrons, making more NADH.
So we're doubling down on NADH. This is exciting.
And the enzyme that makes this happen is called alpha-ketoglutarate dehydrogenase.
Another long one. So are we making any ATP yet?
We're getting there. Step 5. This is where we see our first direct energy payoff.
Finally. Let's hear it.
In this step, a phosphate group replaces CoA, and that creates a high-energy bond.
Okay, yeah.
That energy is used to make GTP or ATP, depending on the type of cell.
So some cells make GTP and some make ATP.
Exactly. It depends on what the cell needs.
Wow, it's like the cell knows exactly what kind of energy it needs.
It's pretty amazing, isn't it? And the enzyme that makes this happen is called succinate thiokinase.
Succinate thiokinase.
That's
Okay,
right.
got it. And what about the molecule? What's it called now?
It's now a 4-carbon molecule called succinate.
Succinate. All right, what's next?
Step 6. Another energy-generating step, but this time a different electron carrier enters the scene.
The new player, huh?
Yep. Succinate gets converted to fumarate.
Okay.
And during that conversion, a molecule called FAD+ picks up the electrons and becomes FADH2.
FADH2, is that like NADH?
Yeah, it's another electron shuttle. It's going to carry those electrons to the electron transport chain.
Okay, got it. So What's the enzyme this time?
This is where succinate dehydrogenase comes back into play.
Oh, yeah, the one embedded in the membrane.
Exactly. Perfectly positioned to pass off those electrons.
Very efficient. Okay, step 7. What happens now?
This one's pretty simple. It's a hydration reaction. Water gets added to fumarate to make malate.
Hydration. So we're giving the molecule a drink.
You could say that. And the enzyme that helps with that is fumarase.
Fumarase.
Yeah.
Okay, we're almost there. Last step 8. What happens?
This is where we come full circle.
Full circle.
Malate gets oxidized, it loses electrons, and NAD plus picks them up to make another NADH.
Okay, so what do we end up with?
We end up with oxaloacetate.
Wait a minute. Oxaloacetate, that's what we started with.
That's the beauty of it. It's a closed loop. The final product regenerates the starting molecule.
So it can just keep going and going.
Exactly. Continuous energy production. And the enzyme that makes this final step happen is malate dehydrogenase.
Malate dehydrogenase. Phew, we made it through all 8 steps. We met all these cool enzymes.
And we generated a bunch of energy carriers, NADH and FADH2.
So what happens next? Where do those energy carriers go? I'm on the edge of my seat.
Well, that's the story for part 2 of our deep dive. You'll have to wait to find out what happens to NADH and FADH2 in the electron transport chain. So last time we were talking about all those energy carriers, the NADH and FADH2.
Yeah, they were like all charged up and ready to go.
Right. And they're headed to the electron transport chain.
The electron transport chain sounds intense.
It is pretty amazing. It's where the real magic happens. The bulk of the ATP is made.
Okay, I'm ready. Tell me everything.
So picture this, the inner mitochondrial membrane where our friend succinate dehydrogenase is hanging out.
Okay, I remember that guy.
Well, that membrane, it's also home to a bunch of protein complexes.
Protein complexes.
Yeah, and these complexes, they form the electron transport chain.
So it's a chain of proteins.
Exactly. And NADH and FADH2, they arrive at this chain carrying their electrons.
Like they're carrying precious cargo.
That's a good way to think about it. They hand off these electrons to the first complex in the chain.
Okay, so the electrons are moving from complex to complex. What happens then?
As the electrons move along the chain, they release energy.
Okay, and then?
And that energy, it's used to pump protons across the membrane.
Protons? What are those?
Protons are just hydrogen ions. They're positively charged.
Okay, so why are we pumping protons? What's the point of that?
This is where it gets really cool. By pumping protons, you create a gradient, a difference in concentration.
So more protons on one side of the membrane than the other.
Exactly, and that creates potential energy.
Potential energy. Like a dam holding back water.
That's a great analogy. And just like water flowing through a dam can generate power, so can those protons flowing back across the membrane.
Okay, so how do they get back across?
They flow back through a special enzyme called ATP synthase.
ATP synthase, that sounds important.
It is. You can think of it like a turbine.
The turbine.
Yeah, as the protons flow through ATP synthase, it spins, and that spinning motion it's used to make ATP.
Wow, so it's like a little machine that's powered by protons.
Exactly. It takes ADP and converts it back into ATP. It's like recharging a battery.
So the Krebs cycle makes NADH and FADH2, which deliver electrons to the electron transport chain, and those electrons power the proton pump, and then those protons, they flow through ATP synthase to make ATP.
You got it. That's the whole process in a nutshell.
Wow, it's amazing how it all works together.
It is pretty incredible, isn't it?
But what happens to the electrons at the end of the chain? They can't just keep going forever, can they?
They need a final destination, and that destination is oxygen.
Oxygen. Like the air we breathe.
Exactly. Oxygen is the final electron acceptor.
Wow, so that's why we need to breathe.
You got it. Oxygen combines with those electrons and some protons to make water.
So it's all connected. The food we eat, the air we breathe, it all comes together in this amazing process.
It really does. It's the foundation of life itself.
Okay, we've talked about the Krebs cycle, the electron transport chain, and oxygen, but how much ATP are we actually making from all of this?
Well, from one molecule of glucose, you can make up to 36 ATP.
36. That's a lot of energy.
It is. And that energy, it powers everything we do, from thinking to moving to just staying alive.
This has been an incredible journey. I feel like I have a whole new understanding of how my body works.
I'm glad to hear that. But there's one more thing I wanted to tell you about mitochondria.
Oh, what's that?
Did you know that mitochondria, they weren't always part of our cells?
Wait, what? You're kidding? Like,
I'm serious. Billions of years ago, they were actually free-living bacteria.
bacteria living inside our cells. That's crazy.
I know, right? It sounds like science fiction, but it's true.
So these little mitochondria, they were just swimming around on their own.
Yeah, billions of years ago, they were free-living bacteria.
And then what happened? How do they get inside our cell?
Through a process called endosymbiosis.
Endosymbiosis, that's a big word.
It means one cell engulfed another cell.
Engulfed. Like swallowed it.
Pretty much, yeah. But instead of digesting the bacteria, the cell decided to keep it around.
Why would it do that?
Well, it turns out that was a really smart move. The bacteria were really good at making energy.
And the cell needed energy.
Exactly. So they formed a partnership. The cell provided protection and nutrients, and the bacteria provided energy.
So it was a win-win situation.
Exactly. And over time, that partnership became so strong that the bacteria became a permanent part of the cell.
And that's how we ended up with mitochondria.
That's right. It's pretty amazing, isn't it? These ancient bacteria are now essential parts of our cells.
Yeah, so we're all part bacteria.
In a way, yes. And you know what's really cool?
Right.
Mitochondria, they still have their own DNA separate from the cell's DNA.
So they're like little mini cells within our cells.
Exactly. And their structure, it still looks a lot like bacteria. It's a reminder of their evolutionary history.
Wow. It's mind-blowing to think about all the things that had to happen for us to be here today.
It really is. And all thanks to these little powerhouses, these mitochondria.
So we've gone from food to energy to ancient bacteria. It's been quite a journey.
It has. And it's important to remember that all of this isn't just textbook knowledge.
What do you mean?
Understanding how energy is made in our cells, it has real-world applications.
Oh, yeah, like what?
Well, in medicine, for example.
Medicine.
Yeah, knowing how mitochondria work helps us understand diseases like diabetes and cancer.
Wow, so it's not just about memorizing facts. It's about using that knowledge to help people.
Exactly. And it's not just medicine. It's also important for things like sports science and nutrition.
So learning about the Krebs cycle, it can actually make us healthier and perform better.
Absolutely. It helps us understand how our bodies use energy and how we can optimize that process.
This has been an incredible deep dive. I feel like I've learned so much.
I'm glad to hear that. But there's always more to learn. Science is a never-ending journey.
That's true. And who knows, maybe one of our listeners will be inspired to make their own discoveries about the Krebs cycle.
That would be amazing.
Yeah.
So to all our listeners out there, keep exploring, keep asking questions, and never stop learning.
Well said. That's all the time we have for today, but we'll be back soon with another deep dive into the fascinating world of science. Until then, stay curious.